Welcome to EPG Pathashala. My name is Dugirala Vasanta. I am on the teaching faculty of the Department of Linguistics at Osmania University. Today's lecture relates to course 10, module 27 of neurolinguistics part of the psychoneurolinguistics course. The title of the lecture is Hemodynamic Methods Used in Neurolinguistic Research. The objective is to describe only two techniques, two or three techniques which are called hemodynamic. The term hemodynamic basically means that the blood flow, monitoring of the blood which goes to different areas of the brain when we are engaged in different cognitive functions. Which part is participating in that cognitive function is studied by monitoring the amount of blood goes to that area. Blood carries oxygen, so the amount of oxygenated blood is what is measured. So the techniques, if you want to trace hemodynamic techniques historically, started with X-rays, then CAT scan, then PET scan, then MRI, then fMRI in that order as shown in the picture next. So you see originally it is photography, which is X-rays, CAT, PET and MRI pictures are shown here, but I will say a little bit more about these technical aspects of it. For details about applications in relation to language, you will have to read the e-text. So let's start with CAT scan. It's actually computerized axial tomography. That's what the expansion of CAT. It's a non-invasive static recording which generates 3D images of the internal structures of the brain, and it was used in the 70s. CAT makes use of X-rays, X-ray beams rotating around participant's head. Once the X-ray passes through the skull and into the brain, it is converted into light sensitive crystal detectors to electronic signals, which then get transmitted to the scanner's computer. Using mathematical techniques, the difference between total radiation emitted and the amount of radiation hitting the detectors is calculated in order to assess where and how much of the X-rays are absorbed. If there is some tumor, if there is something, there is a block, X-rays won't go through. So that's what shows up on the CAT image. Next picture shows you can see the typical scanner, CAT scanner, which some of you may have actually seen in your neurology clinic. So you are made to lie on that scanner, and you go. You go inside, and X-rays are given all around the brain, and the image looks something like this. Again, please see on the screen. You can see that there is a dense area, there is a black area, and there is a small arrow pointing to that area. So maybe that area, maybe it could be a clot. So that's how CAT image shows that X-rays are not absorbed equally in different areas where it was passed. So the difference is what is calculated mathematically, and then they try to locate the problem in that area. But there are some limitations to CAT scan. Because it involves radiation, it's not safe to use with children or pregnant women. And it is suitable for detecting internal injuries of in not only brain, lung and other chest abnormalities and all that, but it has very limited spatial resolution, meaning it cannot identify problems in deep-seated problems. The spatial resolution is very limited. So we need to look at better methods with better spatial resolution. So let's look at the next technique called PET, P-E-T-T. P-E-T-T basically means positron emission transaxial tomography. This uses, instead of X-rays, it uses a radioactive tracer called FDG, which shows difference between healthy tissue and diseased tissue. Basically, PET offers a little more functional and physiological information than CAT. It can detect changes in physiological process in the brain within a time span of 30 to 45 minutes. So obviously the temporal resolution is not all that good, but better than CAT. Using PET, you can create images of glucose metabolism as we engage in cognitive functions, higher mental functions. As you will see on the screen next, basically on the screen you will see 4 images. In the leftmost corner, the first image is showing what happens in the left hemisphere when you are hearing words. Notice the yellow, orange, red indicates maximum activity in that area. When you are hearing words, that maximum activity is in the superior temporal lobe. Alright. On the other hand, the image next to it, to the right side, when you are seeing words, it is more towards the visual cortex in the occipital lobe, not temporal. So, temporal lobe is involved in hearing words, occipital lobe is involved in seeing words, and the third image, bottom left, you see speaking words. When we are speaking, you know, you have studied the earlier modules in neurolinguistics. You know that Broca's area sends signals to motor cortex. See, the red area is in the motor cortex, which is in the frontal lobe. So, frontal lobe, there is more activity, more glucose being carried by the blood, and that is being monitored. The fourth image on the bottom right is about generating words. What it means is, while you are in the scanner, they will show you a word, maybe they will show you a noun and ask you to generate a verb, or they show you a verb and ask you to generate a noun. So, creating a word involves, again near Broca's area in the frontal lobe and a little bit more higher area, as you can see in the picture. So, this is how PET images are monitoring the changes that are happening in the glucose metabolism, which is like monitoring the blood flow and all that. Okay. PET has been used also to compare normal brain tissue and metabolism in normal brains as opposed to Alzheimer's disease, which is an old age progressive neurological disease which comes in the old age. You see the normal brain, the color coding is very different. There is more red and less blue shades in the PET scan. Then early Alzheimer's disease is the middle brain picture, PET picture. You see there is the red is decreased somewhat, the blue is increased. Now you see the rightmost picture, it is completely blue, there is hardly any red color there. So it is showing that something is happening, the cortical areas are either dying or shrinking, because Alzheimer's disease causes certain kind of neurofibrillary, like there are some knots and some structural changes also happen as the disease progresses, and that will have an impact on memory and language, and that can be monitored by comparing a PET scan of normal person with a PET scan of early Alzheimer's and a advanced Alzheimer's, as these images clearly show. But of course, there are limitations to PET scan also. Small movements will not affect images on the PET as they do in CAT, so that is an advantage. But PET is more expensive than CAT. PET scan costs you more, much more. And also a lot of pre-processing of the data is required in PET scan to correct for random coincidences and estimation and subtraction errors and things like that. So, they have of course modified the PET scan into the next level, which is called single photon emission computed tomography, or SPECT, S-P-E-C-T for short. This is a later development. It uses CT technology with radioactive tracer, but in this case not FTG, but iodine 123, which I believe is cheaper than FTG. This is introduced into the bloodstream, and it is much cheaper than PET. SPECT is much cheaper than PET, and it is useful for detecting deep brain injuries also. The next test is called magnetic resonance imaging or MRI. In MRI, a strong magnetic field and radio waves are used to produce detailed images of the inside of the brain. Of course, you can also look at the bones and heart and lungs and whole body. You can use MRI to see what is happening in different organs in the body. But since we are in neurolinguistics, we are more interested in brain. I may keep saying brain, but it can be used for studying organs, internal organs and the rest of the body. MRI scans do not involve exposing body to X-ray radiation and therefore they are very safe. You can use it with children and pregnant women also. But only problem is MRI cannot be done on people who have implants of any kind, like pacemaker, heart pacemaker. If you have, it's a metal and you cannot have an MRI on those people. Let's spend a few minutes trying to learn about how MRI works. So basically, human body is made up of water molecules which contain hydrogen and oxygen atoms. Each hydrogen atom also has a smaller particle called proton. Protons behave like tiny magnets and are sensitive to magnetic fields. When you are inside a scanner filled with magnetic field, the scanner's magnet creates powerful magnetic field and you are pushed into that field. So all the protons in your body line up in the same direction. Then short bursts of radio waves are sent to different parts of the body, knocking out the protons out of their original alignment. And these differences are what are monitored on the computer. So when the radio waves are turned off, the protons realign again. And in the process send out radio signals which are picked up by receivers. So these signals provide information about the location of the protons and distinguish different types of tissues in the body. Which kind of tissue, what kind of protons and how are they behaving? Are they behaving normally as expected? Or is there a block there? Is there a tumor there? Or is there a tear there? Whatever. So basically originally MRI was used as a structural to understand changes in the structure of the human body, internal organs. So this is called structural MRI. It provides detailed images of the brain and body as if we are looking through a microscope. See then the next picture on the screen, you just see the scanner. It looks very much like the CAT scan you have shown, but in this there is a very powerful magnet. In MRI, they use a magnet, superconducting type, and this magnet is an electromagnet made of superconducting wire. Superconducting wire has resistance close to zero when it is cooled to a zero temperature. Once current flows in the coil of this magnet, it will continue to flow as long as the coil is kept at the liquid helium temperature. So basically MRI is used to detect anatomical abnormalities which you cannot easily see, right? And it is good at identifying structural changes in the body about shape, size, integrity of grey matter or even white matter, little bit, okay. But MRI cannot provide information about function. Structural MRI can only talk about size, shape and integrity of structures. Therefore, they have gone on and ahead and developed what is known as functional MRI. Let's look at that next. So, fMRI basically means functional magnetic resonance imaging. It is, what it does is it measures metabolic changes in the brain associated with specific cognitive functions including language. In an fMRI scan, participants will tap the activities can be either language or non-language. So, the examiner might say after you were put into the scan, they might say just keep tapping your fingers. So, the participants may be asked to tap fingers and then they see which part of the brain there is activity, more activity. They might be asked to purse the lips like that, or asked to read, or asked to view pictures, or listen to speech. So, these are functions. When you are performing these functions, what happens to the metabolic activity within the brain Metabolic activity basically means again where is blood going, most blood flowing, or how much of that is highly oxygenated, how much is depleted from this kind of activity is what is monitored in fMRI. So, when there is an activity, the metabolic activity increases, okay, in the area which is responsible for that activity. So, if you are seeing a picture, occipital lobe, most blood goes to the occipital lobe. You have already seen the PET pictures earlier. If you are hearing, it goes to the Wernicke's area and primary auditory area, like that. So, this causes when the blood flows to a particular area, extra oxygen is indicated. I mean, that area gets that blood has lot more oxygen in it, and that is what gets reflected in the MRI image. So, this technique measures amount of oxygen in the blood in a tiny region of the brain. These regions are called Vox cells. Okay. Neural activity uses oxygen in blood vessels. Neural activity uses up oxygen and the blood vessels respond by providing more oxygenated blood to that local region. If you continue to engage in that activity, the blood vessels push more blood, oxygenated blood. Thus, a change in amount of oxygen in the blood is measured in fMRI, and this is taken as a reflection of the amount of local neural activity. Look at the picture on the screen next. If you carefully look at the first picture and count the number of red circles, that is the neural activity in the basal state when you are not doing any activity. Let's count. There are 4, 5, 6, 7 red dots indicating that much of oxygen is there in a basal state. Then you are made to do some activity, activated state. The next one is showing activated state. So, you are involved in some activity, which means the blood should increase, the oxygen amount of oxygenated blood should increase. Now, you count the number of red circles, 4, 6, 8, 9, 10, 11, it is increased. So, more blood is being carried to that area which is performing a particular increased flow. So, basically what they are saying, you can look at the screen more leisurely and study it carefully to understand that in fMRI, neural activity increases blood flow. When blood flow increases, oxygen level increases, that changes the signal, MRI signal, and that is what is captured in the computer and interpreted. So, you may want to know what is the advantage of fMRI over PET. fMRI can register a large number of measurements of functionally significant signals during a short time. Compared to PET, its resolution is good, both spatial and temporal. It uses voxels, that is elementary volume units of the image matrix. Voxels stand for volume units of the image matrix that are assumed to reflect neural response to stimuli. This procedure helps to overcome the effects of strong background noise and sensitivity to other factors like heart rate and head motion, and these are the advantages of fMRI over PET scan. Also, but there are some limitations also. If the participant is very tense or experiencing anxiety because they are inside the scan, it is very closed thing, claustrophobic, then there can be some artifacts introduced into the measurement. Participants who have metal implants, like I mentioned earlier, the heart pacemakers and cochlear implants, cardiac pacemakers and things like, they cannot participate in fMRI also. So, this brings me to the end of my lecture. Let me just quickly summarize. I have tried explaining fundamental information about CAT scan, PET scan, MRI and fMRI. These are the so-called either you can say neuroimaging or more specifically hemodynamic methods used in neurolinguistics. All of them are able to show changes in the deep internal structures of the brain or even functions of the brain. So the early methods like CAT and PET, they don't have as much resolution as MRI and fMRI, and fMRI is much better than MRI in terms of being able to give us information about functional, which area is involved in which kind of cognitive function. We can learn much more from fMRI than MRI. MRI is good at detecting changes structurally, deep-seated wounds or tears or internal hemorrhage or tumors and things like that. So structural MRI and functional MRI are two different things. And so MRI, the other thing is from MRI onwards, MRI and fMRI don't use X-rays which can cause radiation, and therefore we cannot use with children and pregnant women. That problem is not there that much. But if you have any heart implants or cochlear implants and things like that, you don't usually encourage you to go into it. Interferes with the magnetic signals. Therefore, they don't administer these tests to people who are having implants. So the fMRI uses what is known as bold signal. That is, to monitor the blood oxygenated level dependent. BOLD stands for blood oxygenated level dependent signal. In other words, you are monitoring the amount of blood which is rushing to an area which is involved in a particular activity. And when blood rushes like that, there is more oxygen going there. So oxygenated blood as opposed to deoxygenated blood, they differentiate. That is what shows up in the computer image eventually, and they associate that with different motor or cognitive or linguistic functions. If you have any other doubts, please consult the e-text. Thank you.